So where we're picking up now in Bio 121 is the concept of DNA replication. And you might recall that when we're thinking about how it is during interphase, there's G1, S, G2, and then we have mitosis and cytokinesis, P, M, A, and T. During the actual replication piece here, during the DNA synthesis piece, the major enzyme is DNA polymerase, and what's going to do, it's going to make a copy. But the question becomes, how might DNA replication occur? And in early research, there were 3 suggested possible models where you have conservative replication, which is essentially you make 2 strands of something completely new, and you then recombine back in the original structure. Or maybe you make a copy of one and you attach it to the old copy. You make a copy of one and you attach it to the old copy. This would be the semi-conservative model here. And then maybe they intersperse those pieces of information, and that would definitely suggest that this is how you could actually have genetic diversity. And, this could actually drive each of these things you actually see at the phenotype level. Instead, what you're going to see, is, of course, you do a specific experiment. And this is actually a really elegant experiment by Messelson and Stahl, where they looked at E. coli growth in the presence of one of the nitrogen, reviewing for your final exam, nitrogen isotopes, where you actually have the total weight of the isotopic populations for the naturally occurring nitrogen versus nitrogen 15 and nitrogen 14. And what's going to happen is DNA grown in nitrogen 15 is heavier than DNA grown in nitrogen 14. You're going to use this tool that's called an ultracentrifuge. And if you've ever used a washing machine, that's how a centrifuge works, especially on the spin cycle. So what's going to happen is when DNA is grown, you switch the media, after one round of cell division, and the sedimentation of DNA should tell you how this all played out. And how it all plays out, I'll show you in a second, is if the masses were all 1, 15, 15, or 14, 14, or some intermediate between those densities because they're actually being lowered onto this thing that's called cesium chloride. Cesium chloride is a really, really dense salt. You layer the DNA. It's now 15 or 14N, and it should separate by mass, in the centrifuge over a period of time. And that's actually what happens. And what you see is this, is you actually have this semi-conservative model that demonstrates you have roughly an equal amount of 115 and 114 nitrogen, which is then found inside the semi-conservative model. You would have seen different densities if there had been 15-15 or 14-14 or some mixture in between. And this is what was actually used to say that the semi-conservative model was what the mechanism was. So great. We figured out that DNA is the genetic. Think about putting some of these, like the whole story of these experiments together. We've gotten now to this place where we know that DNA replicates, and DNA replicates by making a semi-conservative model. And that information, if you're inside E. coli, can be shared between bacterial cells as a result of either plasmid and or pili movement, but also inside of the cell. But it doesn't address what's actually happening when it's being replicated. And that would take another 10 or 15 years to begin to figure out. But what we do know is, now we know this to be the case, there's a series of polymerases, DNA polymerase 1, DNA polymerase 3, DNA polymerase 2, nowhere in our cartoon here, but we have this mechanism that requires a topoisomerase and this structure that's called a helicase. And to get those 2 strands for the semi-conservative model, you need to unwind the DNA. The topoisomerase releases the tension so the helicase can do the unwinding. And then you decorate each strand with a polymerase. And the polymerase, with the correct primer, will make a 5' to 3' copy. So you'll notice the lagging strand is made in segments here. The leading strand is made as one complete structure that holds the whole thing together. So Mother Nature actually did some pretty cool engineering here. I don't know if you get off on engineering if you think about how it is, how exciting, when you actually design something and it works and it continues to work. But that's what Mother Nature did here. She figured out a way to make these independently small fragments that could then be filled in using a DNA ligase. And this ligase is going to come in handy when we actually think about how things work inside the lab. But ultimately, this is the mechanism that's used. Topoisomerase, helicase, leading strand going 5' to 3', lagging strand going 5' to 3'. Notice that it's actually, we've separated this so we actually create these cassettes. And these cassettes are called Okazaki fragments, and they're ligated into place. So there's two different mechanisms that account for these two new strands, which is kind of cool. Okay? Now, inside prokaryotes, the replication fork is formed when the helicase separates the DNA strand. As I said, the topoisomerase prevents the torsional strain, and then single-stranded binding proteins bind to the single-stranded DNA to prevent the helix from reforming. It's literally blocked. Okay? When you follow the steps through, DNA 1 replaces the RNA primer with DNA. DNA ligase seals the gaps between those Okazaki fragments. So we haven't lied to you. In fact, separating out each of these pieces was Herculean effort, recorded over, I want to say 15 years, but it could be longer than that. But the idea, of course, is there's going to be mistakes that are being made. And DNA polymerase in aggregate also has another responsibility. And that is, if you look here, complementary base pairing, C to G, G to C, travel along, all of a sudden we get to this AT, and a G decides it's going to flop down here inside the A. The DNA polymerase now has the job of making sure that that gets clipped out of there, and now the correct T is added in place, and this continues along. And this explains why, let's see if I can draw this for you without not messing it up. This explains why. So if here's our normal cell cycle, there's G1, here's our S, where this is all occurring, here's our G2, and there's our mitosis again, and cytokinesis. Right, put C here for cytokinesis. That's why it takes so long to actually make this copy. Part of what you're doing as you're going along is you're checking as you're proofreading as you're actually making the new strand. Now, there's another mechanism, it's called mismatched repair, where an incorrectly added base is detected after replication, and you literally have a series of enzymes that can go in there and remove this, okay? And in doing so, it will now allow for the correct nucleotide to actually be put into place, right? So let's see what we're looking at here. GT, right there, notice what it does, comes in, daughter strands, you actually go in, you cleave out the nucleotide, and you put in the correct nucleotide. Okay, likewise, you'd have to do that down here, AC. Okay, so that would actually have to be figured in, okay? And what actually happens, of course, is pauses the entire system. So in step 1, we can actually see that DNA polymerase is proofreading. Mismatched repair can actually now go in and correct some mistakes that might have been missed. And then nucleotide excision, specifically with thymine dimers. Right? We actually use this mechanism to instigate dimer formation inside of DNA when we actually want to arrest the cells from growing inside the lab, right? Hit them with UV, causes dimerization. And then what happens, of course, is now you can actually repair this, and nucleotide excision will occur where you take this entire segment out and then replace it with non-dimerized structures. Okay. So the replication potentially shortens the end of the eukaryotic chromosomes. This is one of the consequences, right? And this actually is Nobel Prize work with Elizabeth Blackburn, 2009. And what she did is she said, hey, if we keep making copies, what's going to happen is the ends of linear chromosomes are maintained because otherwise they'd be unstable at the end. And what happens is telomerase comes in and says, look, we need to cause the complementary strand to be repaired. And you're like, okay, that's not cool at all as far as I'm concerned. But the reality is this. It turns out that telomerase turns out to be a timing mechanism for the stability of your genetic material inside of your cells. And that's where the real work is going to happen in the future. There are many researchers at this point in time that are looking for ways to stabilize or prolong telomerase activity so that the integrity of your chromosomes does not succumb to the aging process. Okay? Literally, millions of dollars are being put into this stuff. And here's Elizabeth right here. Now, what happens when we actually have uncorrected replications that will result in mutations, right? Now we actually know what causes problems. Because we can sequence, because we know how things are being transcribed or replicated in this case, errors occur if uncorrected mutations are completed. Okay, they're not fixed. A silent point mutation here, look, ACG, GTA, CCC, TAC. Okay, notice this gives us serine, valine, proline, tyrosine. Serine, valine, proline, tyrosine. But find the mistake inside the story. When you look at GTA, notice it's GTT, and this wobble codon, this wobble nucleotide here, in this 3-nucleotide codon sequence or coding sequence, right? It created a silent mutation. No problems. A missense mutation results in an amino acid substitution. Okay, where serine, valine, proline, tyrosine, serine, valine, threonine, tyrosine. This is significant. Depending upon which mutation is put in there, in this sense, if this is a conservative substitution, there might be no problem, meaning the grouping of amino acids. Why we consider that to be important will be something in a minute we consider. Nonsense mutations will substitute a stop codon in for an amino acid. And that's what means no nonsense. We're not mucking around here. Serine, valine, proline, tyrosine. Notice what actually happened here. TAC was supposed to be tyrosine. Slap that G in there. TAG is now a stop signal. And whatever was supposed to happen after here is not going to happen. And that's all there is to it. And that can be a lethal event. The fact that they're found means that in many instances they are not lethal. It means you've made some population of some protein that is just behaving inappropriately. Now, friendship mutations. Mutations can lead to protein sequences that are encoded in a slightly different way. Okay? Serine, valine, proline, tyrosine, serine, valine, leucine, leucine. Okay? Now, this could be a shift in the reading frame or an insertion of a codon. And what's going to happen is you're going to notice that whatever's happened here, there's some misread that caused the shift to occur, and that shift then gives us 2 new amino acids, and that gets us back to up here. What type of mutation did it occur, and did it actually turn out to be a disease-inducing or lethal event? So when you're thinking about what else can happen, uncorrected replication errors can also cause chromosomal mutations. So if you actually have some place where you've increased the likelihood of the translocation events that occurred back in the previous chapter, okay, where inversions, fusions, and duplications can occur. But here's something interesting. So far, I've described all of these things as being possibly disease-inducing or possibly lethal. Another real possibility for mutations is that mutation derives the increased activity of whatever that protein may be. It could be a mutation actually becomes a selected advantage in that environment. And this is something that many people simply miss when they're thinking about what happens here. This is one of the ways I actually teach making mistakes as being a good thing. It's going to sound horrible, but you want to make mistakes. You want to be the person who's relaxed enough who can say, look, I'm going to make a mistake. I'm going to learn something from it. And this is the learning piece. Maybe in some cases it's lethal. I don't know. If it was lethal all the time, none of us would be here. Most of the time, it's not lethal. And sometimes it actually means you've created something better than was there before. I want you to go take a look at this. Take a look at the Neanderthal genome. It's a cute little story, about how it is we thought that these genomes were, non-existent anymore. Well, number one, that's not the case. And number two, of course, we mix genomes together and there are things that happen as a result of that. And thinking about what a Neanderthal actually means, that we have to change the way we think about that as well. So I put this in here So you guys hopefully will enjoy that. So what's the distinction then, in this last couple of minutes here about what it means to be a eukaryote versus a prokaryote? Well, inside our prokaryote, most of the work we've spoken about today has been done at the prokaryotic level because the complexity is just simply easier to work with. And that structure is called a nucleoid structure, and everything that happens inside of a bacteria happens inside the cytosol of that. Whereas inside of our eukaryotic cell, over the course of 500 million years of evolution, you can see the complexity. And one thing I want to point out to you, I know our textbook hasn't really stressed it yet, we may have the genetic material for the day-to-day functioning inside of these structures here for that cell. But remember, 380 million years ago, we picked up these guys, and these are mitochondria. And, I haven't reminded you about this in a while, but those actually have their own genome as well. And when we last spoke about them, I was saying signals come from the mitochondria because it has its own genome, it has its own responsibilities. Signals come from the nucleus. If things are going sideways inside of a cell, those are instigation. Those are signals for apoptosis to occur. So even though we are speaking about how this works, if you think about maybe where the genome is located inside of a mitochondria here, you'll see it functions more like a bacteria than it does a eukaryotic cell. Before we finish, we will finish momentarily, right? I want you to think about how this is actually structured. This is what you understand. This is what you've seen. What we want to do is we want to make sure way back when we, inside chapter 2 or 3, when we're looking at the macromolecules, I said we used an octet of histone proteins to create those nucleosomes. And now those nucleosomes are going to create the solenoid shape. And that solenoid shape is further looped back and forth by a series of scaffolding proteins. And this is actually how we make condensed chromosomes. This is what you're looking at. P-arm, Q-arm of a duplicated chromosome that actually has, say, alleles of that material on either chromosome arm. And how now do we switch between our sequencing processes? Here is reading the code for dideoxy Sanger sequencing, right? So we don't use this now. Okay, you can. This is labeled dideoxy. And what you can see here is when you actually DDT, DDG, DDA, these are all enzymatic reactions. They get to a specific distance. When they stop there, they literally intercalate a dye, and that dye can be excited, and you now collect each of these peaks, and this is actually how you sequence the genome. So I had you go look at that Sanger sequencing movie. Maybe you understand it a little bit better now, but the point is we actually have things that go a lot more faster than this. Is that even an English sentence? I'm sorry. It can go a lot more quickly than this. Okay? And now what's going to happen, we can use the sequencing readout shown here as a laser scanner using much more rapid systems. And when you actually get to, if you're taking the bioinformatics class with me, each piece of those sets of data are generated in a mechanism that doesn't look like this at all. In fact, the engineering to understand how sequencing works here is really simple. That's why you start with the Sanger sequencing system, and then you move over to the more complicated systems. Lastly, I want you to see that we can move genetic information around. On the left, far left, and the far right image here, I guess it's just a gel. The far right image, far left and far right. If we were thinking about humans, this would be the right side of the human and the left side of the human if they're your patient. But you can cut fragments of DNA to resolve a specific distance. And I sort of introduced this idea to you yesterday, or in the cyclins lecture. We were talking about how we separate things by their mass. What happens is you cover these with a soap, okay? And that soap in an electrical field passing through a gel separates materials by mass. The smaller fragments move faster and the larger fragments move smaller, and you'll see how we can use this inside lab. Okay, literally where we can cut fragments out. Remember that DNA ligase I spoke about a few minutes ago? That becomes very important in figuring out how we can now just ligate materials together. Okay.